Hello, and thank you for watching. My name is Max Harman, and I'm a rising senior at Kansas State University studying biochemistry and global food systems leadership with a minor in agronomy. This summer, I was hosted in the lab of Dr. Robert Van Buren at Michigan State University and mentored by him and fourth-year PhD student Jeremy Pardo. Being from Kansas, the #1 sorghum producing state, I was excited when I heard that my research project this summer was focusing on investigating the basis for this important cereal crop's drought tolerance. My research has been focused around analyzing data collected during and after a naturally occurring drought event during the 2020 growing season. After 2 weeks of drought, physiology, phenotypic, and expression data were allocated in triplicate across a trial field of 25 genotypes of sorghum, representing the Sorghum Association panel. The same traits were measured again 3 days later following a heavy rain. After the RNA-seq expression data had been sequenced, processed, and quantified, I used the R package DE-seq2 to analyze which genes were up or down regulated in each genotype's drought condition compared to its well-watered condition. I then used the TOPCO package to enrich the differentially expressed gene lists with any gene ontology terms that they had been identified with. Finally, all of the expression data was used in several predictive modeling methods to see if they could accurately separate the drought condition from the well-watered condition. Displayed in figures 3 and 4, the drought physiology data confirms that the plants were experiencing mild drought stress, shown by the significant increase in relative water content following the rain. Interestingly, 4 of the genotypes did not recover as well as the rest. Figure 4 shows the measures of photosynthetic function, linear electron flow, and phi2, which is the proportion of the useful light energy that makes its way into photosystem 2. There is no significant distinction between the drought and well-watered conditions here, showing that the plants were not to the point of drought stress that they gathered damage to their photosynthetic pathways. Figures 5a and 5b show the distribution of up and down regulated genes across the number of genotypes that they were highly differentially expressed in. There were no genes that were highly up or down regulated across all 25 genomes. An important note for this is that this trial and analysis had a cutoff for genes that were differentially expressed at less than a 2.0 log 2 fold chain. This may explain the lack of common expression patterns as an issue in degree. Moving down through figures 6a and 6b, Go terms are labels annotated to genes describing likely functions, mostly via comparison to a well-studied model like Arabidopsis. Go terms are split into 3 broad aspects: molecular function, cellular component, and biological process. And this Go enrichment was run using a molecular function annotation courtesy of Dr. Chad Niederhuth's lab. With future work, we hope to utilize a biological processes annotation to potentially identify lower order, more specific associations. Finally, predictive modeling results were able to be collected with the help of Jeremy Pardo. Figure 6a shows a graphical output of a principal component analysis that appears to successfully separate transcription data from the well-watered. The modeling method below also mostly has high degrees of success differentiating the sorghum's conditions. This is at odds with the apparent lack of uniform markers in the differentially expressed gene analysis, but this may be due to the highly selective 2.0 log 2 fold change filter. Conclusions from this work were that there's a great amount of individuality in high differentially expression genes between all of the genotypes, in this case of mild drought stress. In the future, I hope to rerun the DE-seq analysis and visualize all of the differentially expressed genes with a smaller degree of change and eliminate that 2.0 threshold. I also hope to find either a publicly available gene annotation for the biological process sorghum Go terms, or make my own using BLAST. Finally, I hope to integrate the physiology and outcome data from the sorghum into predictive models to try and predict outcomes and the best drought tolerance mechanisms. I would like to very much thank Jeremy Pardo, Dr. Robert Van Buren, and Dr. Van Buren's lab for hosting and mentoring me for this project, as well as Dr. Addie Thompson's lab for the collection of the data used and input on the project's direction. This work was made possible by a grant from the National Science Foundation, as well as funds from MSU's Plant Resilience Institute. Thank you very much for watching.